Hello. This is lesson 5 of the biology course that I've been working on. I decided to rename it to biology course because, it's mainly just about campus biology. And while it can be used for the avenue of USA Biology Olympiad, also it's just pretty good for general biology too. So yeah. But anyway, this lesson is the last lesson of Unit 1, which I'm pretty happy about. We're finally done with this introductory unit. And so that's one of 8 units done. And then this lesson, it's a really big lesson, but it has a lot of interesting concepts to it, and it definitely lays the foundation for a lot of the later topics that will be discussed. So this lesson is about the structure and function of large biological molecules. And yeah. So the first concept 5.1, is a really short concept. Basically, it's just saying that these large biological molecules are called macromolecules, which are just molecules known for their huge size. And then basically, these molecules are also called polymers. And polymers, we know that poly means multiple, and so a polymer is a long molecule consisting of many smaller parts that are each identical, and those smaller parts are called monomers. And so polymers are made up of multiple monomers. And so, yeah, that's what these large molecules, all of them are. And then, so some of them, these polymers are formed by a process called polymerization. Um, and then they can also be broken down. And these processes, the breaking down and the creation of polymers, are facilitated by enzymes. Now, enzymes are also molecules, macromolecules, that um, basically speed up chemical reactions. And they do this by lowering the energy required to start the reaction. So that's kind of a bit of extra information. You don't actually need to know that, but just know that enzymes speed up reactions. And so there are various different reactions that can form polymers, but the main one is called a dehydration reaction. And so basically, a dehydration reaction is when a um, water is released or lost to form a bond. And then there's also a dehydration reaction is a type of condensation reaction. So a condensation reaction is basically a reaction where a bond is formed for the loss of a molecule. And in dehydration synthesis, or a dehydration reaction, is a form of condensation reaction where the molecule that's lost is water. So dehydration reaction also has an opposite called hydrolysis. And so hydrolysis is when a bond is broken by adding water. So it's just the exact reverse. And this graph here, or this, not graph, sorry, this diagram here is a really good example of it. And then, so these are glucose. And basically, so as you can see, it forms a bond via dehydration synthesis. And so it forms an oxygen bond, or a covalent bond with oxygen, by removing the water. And you can see the reason it does that is because it has an OH and an OH, two hydroxides. One oxygen leaves behind, then the H2O, the two hydrogens, and then the oxygen leave as water. And that forms the covalent bond there. And then you can just add water to it, and then they separate again. So basically, that's just kind of like the main chemical basis for all of these reactions. And then there's also a small bit about how diverse polymers are. I think it kind of makes, it's just kind of like a no duh. Like basically, like just there's just so many different monomers and polymers that they can form just an uncountable number of different variations or different conformations of polymers, and that's why they're so diverse. There are many different types, and we can see that later. Okay, so the next concept is concept about carbohydrates. So basically, carbohydrates are just sugars, and they are what mostly power all life. They provide the energy for life. And then the actual science behind that will be discussed in later lessons, but mainly it's just about how carbohydrates can release energy, and that's how they power things. But basically, the monomers of carbohydrates are also, carbohydrates is a term that's interchangeable with sugars. All carbohydrates are sugars. Well, all sugars are carbohydrates. So basically, carbohydrates, the monomer is monosaccharides, and then the polymer is polysaccharides. That kind of makes sense. So monosaccharides make up the polysaccharides, and essentially, the monosaccharides all are basically just they're all made up of either hydrogen, oxygen, or carbon. And so I don't have, let me see if I have, oh yeah, I don't have like a graph of like what the chemical structure looks like, but mainly it's just a chemical skeleton and, oh no, sorry, not chemical skeleton, a carbon skeleton, um, a carbon backbone. And so basically, um, in all monosaccharides, there's a carbonyl group, which is, if we remember back to it, it's a carbon double bonded to an oxygen, and there's also multiple hydroxide groups, which we can basically see here. This is it. This is a glucose. These are glucoses, and basically there are many different monosaccharides. So there are three types. There's a triose, which is a 3-carbon sugar. There's a pentose, which is a 5-carbon sugar, and then there's a hexose, which is a 6-carbon sugar. So most of the sugars that we eat, like glucose, galactose, and fructose, are all hexoses, and then pentoses are like ribose. Those are the stuff, ribose and deoxyribose. That's the stuff that is in DNA, which we'll talk about later. And then trioses are 3-carbon sugars, and then they break down glucose during glycolysis. And so um, there's also a type of polysaccharide called a disaccharide. So these disaccharides are just made out of 2 monosaccharides. They're 2 things bonded, 2 sugars bonded together. And then um, they are bonded by what's called a glycosidic linkage. So a glycosidic linkage is a covalent bond formed between 2 monosaccharides via a dehydration reaction. This is literally this exact thing here. This is literally a glycosidic reaction. So it's good that I referenced it earlier. So yeah, that's a glycosidic linkage. Just remember that. And basically, these sugars are important in life because they all, like I said, they power everything. That's what we seek to gain when we eat food and stuff like that. It's what forms as our energy. And then they can be used as storage in fat. They just store energy and stuff like that. And so there are actually 2 different types of glucose structures. I think this applies to actually all hexoses, all 6-carbon sugars, because there's a alpha glucose form and it's a beta glucose form. So the alpha glucose form is basically if we kind of remember back to the cis-trans structure of things from the earlier lesson, notice how trans, they were diagonal, and if it was cis, then they were parallel. So alpha glucose is the cis structure, where these 2, the hydroxides and then the hydrogens are parallel to each other. But if you have a trans structure, then they're diagonal to each other, as we can see. So yeah, and basically the interesting thing about alpha glucose and beta glucose is that their carbon skeleton structure looks the exact same, but if you look at the 6-carbon ring structure, so which is what I have here, then you'll be able to see the difference. And because of this difference, it changes a lot of ways how glucose interacts with other molecules. As we know, the main theme is that structure defines function in all biology. Okay, I'm gonna go back to disaccharides. So there are many different important types of disaccharides that you don't need to know them, but they're just good to know. There's lactose, which is the sugar in milk, and then there's also a form of sucrose in plants. That's also a disaccharide. As you guys can tell, all sugars have the root of ose. So yeah, if you hear ose, it's probably referring to a sugar. And yeah. Also remember that all disaccharides are formed via a glycosidic linkage formed via dehydration synthesis. So now on the topic of polysaccharides, the things I was talking about before were all mono or disaccharides, like glucose, galactose, fructose, ribose. But for polysaccharides, these are basically just multiple monosaccharides bonded together. As we know, they're polymers. And so they're all joined by glycosidic linkages. And so these polysaccharides are what we see as like sugar. There's also starch, which is in plants. And there's also chitin or chitin. I don't know how to pronounce it, sorry. Chitin in like cell walls of plants. And there's also cellulose, which is another pretty important one within cellulose is the one in plant cells. Chitin is the one in I think fungi. Oh shoot, no, sorry. It's used in arthropods for exoskeletons. So basically, for polysaccharides, there are actually three types of um, branching that you can have. So these are just the main two ones. So if you have an unbranched polysaccharide, that means that the sugars, the monosaccharides, are all in a straight line. They're all formed by one glycosidic linkage like this, and then it's just a straight line like this. If you have branched polysaccharides, then they're branched like this. They're bent. The the polysaccharides like bent all over the place. And so there's also kind of a type that's like mid branched. It's like branched, but like kinda like not really branched. It's only branched in some places. So some examples, there's amylose. Amylose is unbranched. Amylopectin is kind of branched. Glycogen, which is don't get it mixed up with glucose. Glycogen is extensively branched, so basically it's just really branched, and then cellulose is unbranched. So just keep those in mind. You will be quizzed on that later. I think so. Yeah, I think you will be quizzed on that. Just yeah. And so the branching, it affects like the strength of the sorry, I heard something. It affects the strength of the glucose structure, the chemical structure, and yeah. So, yeah, glycogen is also a polymer of glucose, by the way. That's the name for the polymer of glucose. And amylopectin is the starch, a more complex starch. So yeah, yeah. Remember branching, and then, okay, yeah. Sorry, I'm checking something right now. Okay, yeah. So earlier when I was talking about alpha and beta structures, and how it affects how things interact with other ones, it's pretty important in biology because it affects how some glucose can be digested. For example, it's not glucose, sorry, some hexoses can be digested. For example, cellulose cannot be digested, I think. And then, yeah, that's kind of it. And then these are called insoluble fibers. That's pretty interesting. Yeah, fibers are not digestible, I think. I don't know. I'm sorry. But yeah, I mean, all this information isn't super like important to know. Yeah. Okay, but that's all you need to know for carbohydrates. Okay, for the next concept, it's lipids. So lipids are probably one of my favorite macromolecules. I think they're really interesting, and then there's a lot of different uses for it. So very much like carbohydrates, it's basically just a carbon skeleton. Like so, um, basically lipids are hydrophobic macromolecules that are also called fats. And then they're really significant because they're the only macromolecule that's hydrophobic, and they're made up of fatty acids. So this structure here that I'm pointing out with my mouse is a fatty acid tail. That's the fatty acid. And then this here is a triglyceride structure. So most fats are triglycerides, meaning that they have three glycerides or three glycerols. And a glycerol is also a fatty acid. Yeah. So basically, there are three fatty acids made up of it, and then as you can see, it's all really just a carbon skeleton. It's all made of hydrogen, carbon, and oxygen, and it's pretty similar to carbohydrates, but don't get them mixed up. So yeah, these fats basically lipids are like the fat in your stomach. There's also like the fat that you eat. There's also butter, lard, oils, and yeah, those are all lipids. And then basically, and they're also called esters. So an ester is basically, hold on, let me find it. It's basically a linkage between, it's this linkage here. That's what an ester is. And then the esters are typically associated with lipids because they form the triglyceride like linkage. It's the linkage is the carbonyl with an oxygen, and that's what binds the fatty acid here. This thing, and that's what binds it to the together to form the triglyceride. So, it's also important to note that in triglycerides, not all of the fatty acid units have to be identical. Obviously, there are different types of it. They mainly differ in their type by the length of it. Some of them may have more hydrogens or carbons. And then, so as you can see here, not all of them are the same. Only 2 of them are the same. So yeah. So basically, there's also, this is a pretty important thing to know. So there are saturated fats and there are unsaturated fats. So basically, they're caused by a linkage in their fatty acid tails. No, sorry, not a linkage, a kink in their fatty acid tails. So this kink is formed by a double bond. As you can see here, it's visible here. The double bond between the carbon and the between the carbons. And basically, because of this because of this double bond, it basically causes less hydrogen to be able to bond to the fatty acid tail. And as you can see here, there's less hydrogen here. So also it causes the structure to be bent, which is what we see here. But basically, because there's less hydrogen there, it's unsaturated. It's not, but when there is regular bonds, there's no double bond, then more hydrogens can bond to the structure, and therefore it is saturated with hydrogens. That's where the name comes from. So basically, when there's more hydrogen, a saturated fat is typically solid at room temperature. Butters are saturated fats, and then yeah, and then when you have an unsaturated fat, then it's liquid at room temperature, such as oils and fish fat. Those are all unsaturated fats. And so just remember that unsaturated fats have the double bond and they have a bent structure, and it's caused because of that double bond. It has less hydrogen, so it's not saturated with hydrogen. So yeah, it's a pretty important thing to know. And then so also just know that nearly every double bond in naturally occurring fatty acids are all cis double bonds. So if we remember back to cis double bonds, that just means that they're parallel. I was talking about it earlier. And then yeah. So there's also a thing called hydrogenation. And so that's basically when you add hydrogen to fats to basically synthetically convert unsaturated fats to saturated fats, adding hydrogen to it. And the reason they do this, they only do it in food. The only reason they do this is to basically make them solidify. Like I said earlier, saturated fats are all solid at room temperature. And so that's why like stuff like peanut butter, ketchup, they can separate out into an oil form, but they hydrogenate it to make it not separated out into an oil form. So yeah, I'm gonna drink some water. Wait. So, there's also basically when you hydrogenate fats, there's also a possibility that you create saturated fats, but also an unsaturated fat with trans double bonds. So if you remember what trans double bonds are, they're the diagonal ones. And so these fats with trans double bonds are called trans fats, and they're kind of dangerous because they cause heart disease, and then yeah, they're like cholesterol. I think no, not cholesterol. Sorry, but like they're just really bad for you, and that's why trans fats, yeah, they're not good. So yeah, and here I wrote a note that says trans fats are hydrogenated oils that form unsaturated that. What does that say? Unsaturated trans bonds. Okay, whatever. Okay, yeah, whatever. Anyway, yeah, just know that like trans fats are bad for you. Don't eat them. Um, yeah, I think that's why I think trans fats are the reason why people say synthetic foods are bad for you, because it's purely synthetic, and then because of that, it's unhealthy. But yeah. So another really important thing about lipids is that they actually form phospholipids. So phospholipids, I have it here. It's basically a structure of 2 fatty acids and a phosphate group at the top of it. And so essentially, It's really important because it actually forms the cell membrane that actually surrounds the cells. It forms a cell wall. And so basically, if you didn't have phospholipids, then you wouldn't exist because there wouldn't even be cells. Like no life would exist. And that's why it's so important because of this phospholipid membrane. And so this phospholipid thing is called a phospholipid bilayer. And it's really interesting because we know that the phosphate group is hydrophilic, meaning that it likes water. But we also know that lipids are hydrophobic. And so, because of this, because there's a hydrophobic part and a hydrophilic part, then what happens is the phospholipid bilayer, the hydrophobic parts face each other, the fatty acids face each other, and then the phosphates face outward. The hydrophilic parts face outward, so they can interact with water. And that's why it forms this structure here, where we can see that the lipids face each other and then the phosphates face outward. So, I think it's really cool that they do that. It's kind of similar to how proteins work, which I'll talk about later. But yeah. And then these phospholipids, they do a lot of things that they can prevent things from coming inside the cell, but they also can recognize other proteins or enzymes, and they allow it to exit the cell or enter the cell. It's just pretty cool. And then yeah. So there are also other types of lipids. So steroids are actually a form of lipid, and steroids, I'm not talking about the things that make you buff. I'm talking about testosterone or estrogen or progesterone, and these are all actually sex hormones, but they can also be called steroids. And essentially, they're characterized by a carbon skeleton with 4 rings. So it's formed. There are six. There are 3 6-carbon rings, and then there's also 1 5-carbon rings. And as you can see here, this is basically, I'm not sure which one this is, but this is just recognize the 4 rings. That's how you know that it's a steroid. And then there's also a form of a steroid called cholesterol, and we know cholesterol is bad for you, but also it's actually pretty important for you. You still need cholesterol. And then, but if you have a high level of cholesterol, then it can block up blood vessels, and that's what causes heart attacks. Yeah. So yeah, that's all you need to know about lipids. Just remember all that. Okay, the next concept is proteins, my favorite one. I actually do protein design research at Rice University. But yeah, so I know a lot about proteins already. But yeah, basically, proteins, they're what allow literally everything to happen in your body. Any activity that occurs in your body, it's probably because of proteins. They're really cool because they have there are hundreds and thousands, millions of them, and they all have a unique three-dimensional structure. This is just one of them. They have coils, they have sheets, flat areas, they have long strands. It's just really cool. And then proteins actually account for more than 50% of the dry mass of most cells. So they're quite ubiquitous and they're everywhere. And then some proteins are called catalysts. Basically, they're just chemical agents that speed up chemical reactions. They're enzymes. Enzymes are catalysts. Catalysts are proteins. Enzymes are proteins. And then basically, a protein is formed by amino acids. So amino acids, there are 20 different amino acids, and they are all different. They're all unique, but they all have this one same part of them, which is a protein backbone or an amino acid backbone, and that's what this is. So it's characterized by a asymmetric carbon, which we remember back to the earlier lesson. An asymmetric carbon is a carbon with 4 unique things that it's bonded to. So there's an amino group, and then there's a carboxyl group binding to that alpha carbon along with hydrogen, and then there's a side chain. So the side chain is what makes the amino acids unique. It can be anything. For example, in cysteine, there's a sulfur in the side chain and some other stuff. In proline, there's there's a ring there. It's very interesting. And so, but this amino and carboxyl group are what make proteins unique. And essentially, what happens is these these amino acids, which are also called peptides, are bonded together by a peptide bond to form a polypeptide. And basically, they're bonded by, take a guess, it's dehydration synthesis. So the backbone here is what bonds the amino acids together. So the amino group of one amino acid bonds to the carboxyl group of another amino acid. And so the hydroxyl here, and the hydrogen of the hydroxyl of the carbon of the carboxyl group, and then the hydrogen of the amino group bond together. And what happens is the these two become water, and so as you know, in dehydration synthesis, the water leaves, and so that allows the nitrogen of the amino group and then the carbon of the carboxyl group to bond together, and then boom, you have connected amino acids, and that forms a polypeptide. And so, yeah, I don't know, it's just really cool. I love chemistry so much. It's so cool. Yeah, and then so the polymer of amino acids is called a polypeptide, but just remember that a polypeptide and a protein are not interchangeable. When you talk about a protein, you're referencing the whole three-dimensional structure. So that includes like all of the coils, the chains, the sheets. But when you talk about a polypeptide, you're just talking about the amino acids, the list of amino acids. So don't get them mixed up. I mean, yeah, yeah. So, a peptidic bond is that bond between the carbon and the nitrogen, the carbon of the carboxyl group, and then the nitrogen of the amino group. So yeah, yeah. So, I think that's all you know for that. Also, by the way, the asymmetric carbon at the center of an amino acid backbone is called the alpha carbon. Yeah. So proteins can do a lot of different stuff. They can form as enzymes that speed up reactions. They can kill diseases. They can transport molecules. They can they can react to hormones that cause part of the endocrine system, and they also build stuff like your flesh, your bone, your skin. Also, the hormones that they interact with, those hormones are also proteins. And then they can also do stuff like in plants. We know that the vacuoles store water. Those vacuoles are storage proteins, stuff like that. They also make your muscles, just everything. Yeah. So, there are 20 amino acids, as I stated earlier, and these amino acids are all very unique in their own way. So, let's drink some water. Hold on. So, there are amino acids that are polar, that are hydrophobic, and there are amino acids that are hydrophilic. So, the amino acids that are non-polar are hydrophobic, because as we know, water is polar, and polar goes with polar. So water is only going to interact with things that are polar. And so if you have something that's non-polar, then it becomes hydrophobic. So as you can see, these amino acids with hydrophobic side chains, these are all non-polar. And then these are amino acids with polar side chains, these are all hydrophilic. So yeah. And then the electrically charged ones are also all hydrophilic as well. So basically, technically these special cases also fall into each of these three categories, but I think this chart just separates them. So I'll talk about the special cases first. So cysteine, like I said earlier, it's very special because it's the only amino acid with sulfur. And then if you have glycine special because it's the smallest amino acid. And then proline is special because it's the only amino acid with this 5-ring chain, or 5-ring like thing here. And then that's special because oftentimes when proline is inside a protein, it often causes the protein to have kinks in it, and it has weird bends and stuff like that. And then I know, like, one of my lab partners, she was doing a project, and then one of her proteins that she was testing on had a lot of issues because there's just so much proline inside the protein. And, because it just causes a lot of issues. So, yeah. Anyway, okay, so on this lesson, I'm pretty sure I have a booklet that helps you memorize all 20 amino acids. It's not inherently necessary, but I know that there are professors at universities that force you to memorize them anyway, and stuff like that. And then, I don't know, I think it's just a helpful skill to know and stuff like that. But yeah. So yeah. You don't need to know all these, but just look at them. They're all so complex. But yeah. Okay, so with proteins, there are actually 4 different stages of protein structure. Okay, wait, I think I forgot to mention something. By the way, the amino group and then the carbon group, this is called the C terminus, and then the carboxyl group, this is called the C terminus, and this is called the N terminus. I think it was pretty obvious because there's a C there's a carbon in the carboxyl group, and there's an N for nitrogen in the amino group. But yeah, just know that. Anyway, so there are four different parts of there are four different levels of protein structure. There's the primary structure, the secondary structure, and the tertiary structure, and then the quaternary structure. So technically, in a lot of proteins, there actually isn't a quaternary structure, which I'll explain why later. But I'll start from the very beginning. So our primary structure, it's just that's the polypeptide. It's the linear chain of amino acids, the list of amino acids. And oftentimes, people will just like represent the primary structure with just like, I don't know, A, B, C, D, E, F, G, like just like a list of letters. That's how the PDB does it, the protein database, I mean. And then, so that's the primary structure. The primary structure, yeah, is just a list of letters. Like, that's kind of it. So it gets interesting in the secondary structure. So basically, the secondary structure is when you start getting the three-dimensional things. Basically, like with chemical conformations, it forms different kind of parts, components of the actual full structure. So, like I said, you can have either a sheet, which is like called a beta sheet. As you can see here, it doesn't have to always be three strands. So yeah, or you could have a alpha helix. So this graph shows it better. You can see like how the alpha helix works, and then you can see how the beta sheet works. As you can see, there are hydrogen bonds between all of those. And then here you can see there's so the actual number for an alpha helix is 3.4 amino acids per amino acid turn. That's just a fun fact. But yeah, I mean, sorry, alpha helix turn. So I like alpha helixes more. I think they're pretty cool, but beta sheets are also pretty cool as well. And then basically, the reason that proteins form in such a manner is all about energy. So this is when really the chemistry kind of stuff for biology comes in. Most of like stuff like this is all caused by entropy and energy. Basically, they want to have the most surface area while also having the smallest volume, and that's how it maximizes energy. So that's like why like you can see this in other stuff too. That's why mitochondria is kind of like squished together in there, because it has a lot of surface area but takes up very little space. And so yeah, it's pretty interesting actually. So yeah, that's kind of like the chemical reason why they form like this. Like if you're wondering why they form like this, it's because of energy. It maximizes energy this way. So yeah. The beta sheet is also called a beta Pleiadic sheet. They just mean it's the same thing. So yeah. Anyway, the tertiary structure is where it all comes together. So tertiary structure, in a lot of proteins, this is the final stage. A tertiary structure is just the full two-dimensional shape of a protein or a monomer of a protein. So basically, the beta sheets, the alpha helixes, they all come together. You can also just have polypeptide chains that like connect them all. And yeah, this is a good example of a tertiary structure. So a quaternary structure is just is if a protein has multiple copies of like the same monomer. Yeah, it's kind of interesting. Like you have like the polymer and the monomer with the amino acids, and then the protein. But we can also have like a protein of proteins, like a polymer of a polymer. Get it? I'm sorry, but yeah, this is one example. So this protein here, it has three copies of the same thing that come together. So it's three monomers, and then the actual names for this are trimers. Like trimers, you can also have dimers. You have hexamers, what is it called? Tetramers. It goes all the way up to 23, I think. Like you can have up to 23 like copies. That's all we've discovered so far. And then these things are called oligomers, like oligomeric. Like these parts are called oligomers, and they form a homooligomer, like copies of the same thing. I don't know, I'm just throwing random facts now. But yeah, so basically, the reason I was saying that a lot of proteins don't actually have a quaternary structure is because most proteins, no, not most, but like a lot of proteins don't have like a copy of like parts. Like it's not made of multiple monomers. So yeah, most proteins just like end up like this is like the final stage. For example, this protein here, that's just it. Like that's the monomer, or that's just the final stage. It's not a monomer. But yeah. But yeah, those are the four stages of protein structure. See like this example here, I'm pretty sure that's hemoglobin. Hemoglobin has, it's the stuff in blood. That's what, like, it's what gives blood the red color, stuff like that. And then, hemoglobin, it has four parts, four monomers. It's a tetramer. Tetra is the root of four from Greek. So yeah. There's some also like interesting stuff here. Okay, so if you're wondering how the tertiary structure forms like this, one of the most interesting reasons why the structure forms the way it does is because of hydrophobic interactions. So as I stated earlier, some amino acids are hydrophobic while other ones are hydrophilic. So it's the same thing with, as I was talking about earlier, with the um, what's it called, the fatty acids, the phospholipid bilayer. So basically, some amino acids want to interact with water, they're hydrophilic. Other ones don't want to, and because of that, the parts of the protein structure that are hydrophobic face inward to each other, so they don't have to interact with water. Meanwhile, the ones that are hydrophilic and want to interact with water face outward, and so the protein folds inward on itself. There's a lot of weird like movements to try and get all of the hydrophobic proteins facing each other and away from the outside, while the ones that want to interact with water face the outside. So yeah, and then the shape of a protein can also be caused by disulfide bridges. So disulfides, meaning two sulfides. This is mainly caused by cysteine, as I was talking about earlier. Cysteine is really significant. It's the only one with sulfur in its structure, and because of that, cysteine kind of like defines a lot of the um, three-dimensional structure of a protein. Yeah. Okay. So there are two types of proteins. You have a fibrous protein, which is just like a long strand. This is stuff like collagen and like I think hair keratin. Yeah. And then you can also have globular proteins. So this is a globular protein. This thing here, it's like just globular, like the sound of it. It's just clumped together, stuff like that. And then fibrous, it's like a fiber. It's long strands. So yeah. Okay. So with proteins, there's some more information. So as I was talking about earlier, with hemoglobin, it's the cells in blood, and then no, it's the stuff in blood. It's what gives blood the red color. It's in blood cells, stuff like that. So sickle cell disease is a kind of a genetic disease that directly affects hemoglobin, and then it causes the blood cells, instead of the regular red circles, you get kind of like discolored moon-shaped blood cells, and then that affects a lot of things. For example, the blood cells don't really carry oxygen that well, and then they also have trouble binding to things, and then mainly the blood cells actually move through the blood vessels really difficultly, and then sometimes they can clump together and block your blood vessels, and that's why sickle cell disease is so deadly. And then the one of the important things about sickle cell disease is that it's actually only caused by a single amino acid being off. The sixth amino acid in the structure, glutamate, or I think it's glutamate, or glutamic acid, is replaced by valine. And that one change causes the entire protein structure to misfold, to be completely off, and that causes the entire cell to be off. So it just really shows how every amino acid within the protein is incredibly crucial to how it forms and how it works and stuff like that. So yeah. And then there's also a process called denaturation. So basically, proteins don't always keep their structure the same. If it ever, there's ever a large change in temperature, too much pH, it's either too acidic or too basic, or there's like salt or any other like environmental things are changed, then it can be denatured. And denatured is basically when a protein unfolds, or it just like kind of gives up on itself. Like, look at this. This is denaturation. So you have like a globular shape folded protein, and then it denatures. It unfolds itself into just the long strand. And as we know, structure defines function. So once the structure is messed up, then it's basically unusable. Your proteins fall. And so, it's really interesting to see. Like, for example, like, when you like cook eggs, like in the pan, how to like the white stuff becomes a solid white things. Those are the proteins literally denaturing into, and then it becomes solid. Another thing is like, I don't know, if your finger like touches something hot, then it like blisters up and like turns red, stuff like that. your proteins in your finger are literally denaturing because of the high heat, and then that's what causes that stuff. Um, so like one of the most interesting denaturation is with chloroform. So basically, when a protein interacts with chloroform, the polypeptide chains refold so that the hydrophobic regions that were previously facing inward fold outward so that they can interact with the chloroform because they're only afraid of water. They want to interact with the chloroform. So I think that's pretty interesting. Like it literally goes inside out. So I know that's pretty cool. And then another important thing about denaturation is like heat. Like this is why like really high fevers, like if your fever is like 100 and like I don't know like 10, you're gonna die because it's so hot that your proteins are like they are it's too hot and they misfold and your entire body like just dies. So yeah. Anyway, lastly, there's also with denaturation, there's also another form of just proteins being misfolded, and these misfolded proteins are called prions. So prions are actually really deadly diseases. They can cause really deadly diseases. They cause cystic fibrosis, Alzheimer's, Parkinson's, mad cow disease, and the thing with all these diseases is that they're incurable. The reason for that is because these diseases are called by prions, which like I just said, are misfolded proteins, and they're because they're only just misfolded proteins, your body has a really hard time even noticing that they're actually diseases, and along with that, they're not actually like things you can really kill because they're technically not living. They're just what happens is that these misfolded proteins interact with other regular folded proteins, causes those proteins that are once really good folded to also misfold themselves into the same form, and then what happens is your entire body just kind of like misfolds itself, and then you just die. And it's incurable because well, it's not something you can kill. It's not like a disease you have to kill, and nor is it something that your body can really detect because it just thinks it's like another protein. And that image down here is a misfolded protein. As you can see, it just looks sinister. Like it just looks evil. Yeah. So, it's also why like prions, like whenever there's like I don't know an outbreak of mad cow disease, people want to stay as far away from those proteins as possible because proteins are a universal thing in all organisms. So like that's why humans can also get prion diseases, and then you can transfer it to your I don't know your pets or stuff like that. So yeah, and then so you may be wondering how these three-dimensional structures of proteins even emerged. So they were actually generated by like not generated, but they were discovered by something called X-ray crystallography. So basically, this is when you use light diffraction from an X-ray beam. You shine a beam of X-ray, which is a form of what is it called again? I forgot what it's called. Like photoelectro. I don't know. I forgot what it was. But anyway, X-ray. So, uh, basically, when you shine an X-ray through like a protein, the atoms of a crystallized molecule will cause a specific diffraction of that, and they can kind of like predict or just like know the structure of a 3D model of the molecules in that protein. So yeah. There are also different methods that you can use to find proteins, but X-ray crystallography was, I think, the first one and also the most popular one. I mean, now we just use machine learning models like AlphaFold and R Rosetta. Um, I just do that in there. It's a fact. Like I'm not. This is a fun fact. Like I work in that. But yeah. Um. Oh yeah. One important thing to know is that like some proteins don't actually have like a fully defined three-dimensional structure until they interact with something, and then they form themselves around whatever they're bonding to or whatever they interacted to with, to be like that. So that's when they actually get their defined three-dimensional structure. That's pretty cool. Okay, that's all for proteins. I'm gonna drink some water. Wait. Okay. So, nucleic acids. Basically, nucleic acids are what make up genes. So our DNA, stuff like that, genes. I think at this point, like, I think most people should know what DNA is. But basically, genes are just kind of what make you, you. They make us like us. Um, yeah. Basically, I mean, genes, they're code, their genetic code, their like biological code, and then that's what code us. It mainly codes amino acids and proteins to make proteins. Like it's like kind of like genes are kind of like the instructions for how to make the proteins that your body makes, and then nucleic acids are polymers made up of nucleotides. The nucleotides are the monomers. So yeah. Okay, basically, so there are two types of nucleic acids, DNA and RNA. They kind of like complements of each other. Not complements, sorry. Basically, DNA is the stuff like the genetic instructions, and then RNA controls protein synthesis. And basically, what happens is DNA codes, it makes RNA, and the RNA does a lot of stuff that, we're not going to delve into specifics here, but it's a process called gene expression, and then it basically RNA copies the DNA in a process called transcription, I think, and then another type of RNA reads whatever RNA copied from the DNA in a process called translation, and then that tells it how to make a amino acid structure. So that entire process is called gene expression. So, I think we all know what chromosomes are. They just hold DNA, stuff like that. It's like the stuff in cells that hold DNA. Yeah. Okay, it's actually a lot more information than I thought it would be. The different types of RNA. There's like messenger RNA. That's like what carries the information to other RNA. There's also like rRNA. That's what makes the proteins. rRNA is also called a ribosome. So yeah. I have a graph of I have a graph of gene expression later. I'll explain that later. So basically, nucleic acids are polymers that are called nucleotides. Polynucleotides, and then the monomers are nucleotides. Okay, so a nucleotide is made up of 5 components. Three components, okay? It's what's shown here. Okay, you can have. So first you have a phosphate group. That's what's the backbone that's facing outward. Then you have a pentose sugar. As we discussed earlier in the carbohydrate section, a pentose sugar is a 5-carbon ring, and then, like I said earlier, ribose is one example. And then you have the actual nucleotide, and that's what makes the entire nucleotide, and then that bonds together to form a polynucleotide, and then that's what makes your DNA and RNA. So what you're looking at here is actually just the molecular structure of DNA. As you can see, there's like 2 rings, 2 helixes. This is one helix, this is one helix. So there are 4 types of nitrogenous bases. So the nitrogenous bases are the things that bind to the pentose sugar, and that's what. I'm just some more water. I'm actually so thirsty. Okay. So nitrogenous base. That's like the important part. That's the actual coding part, because the phosphate and the pentose sugar are common. It's like all of the nucleotides have it. Okay. So, I think I misexplained it earlier, so I'll explain it again. So like, polynucleotide is this whole thing. It's like just like all of the nucleotides mixed together. A nucleotide has 3 parts: phosphate group, pentose sugar, and then the nitrogenous base. So there are 4 nitrogenous bases. You have, there are actually 5 actually. So there are there are 2 types of nitrogenous bases. There's purines and pyrimidines. So and then there's 2 purines and there's 3 pyrimidines, and then those are the 5 nitrogenous bases. So as you can see, the purines are adenine and guanine, and purines are characterized by their 2 rings. Pyrimidines are cytosine, thymine, and uracil, and then they're characterized by the fact that they only have 1 ring. And so each purine goes with a unique pyrimidine, and then vice versa. And then basically uracil is only found in RNA, and then adenine is only found in DNA. Speaking of, so DNA is called DNA because of, as we know, deoxyribonucleic acid. RNA is called ribonucleic acid. So hence the name. DNA does not have oxygen in the pentose sugar. So it lacks an oxygen atom, so it's deoxy, right? So anyway, um, for the thing for the structure, okay. So adenine always goes with thymine, and cytosine always goes with guanine. And then in RNA, when you replace adenine with uracil always goes with thymine. So yeah. Um, I'm actually not sure the chemical reason for why uracil replaces um, adenine. I'm actually not super sure. Oh shoot, wait. Shoot. I messed it up. Oh shoot. Wait. Forget everything I just said, okay? So, uracil replaces thymine. And so, adenine always goes with thymine, guanine always goes with cytosine, and in RNA, when uracil replaces thymine, adenine goes with uracil. Okay, yeah, sorry, I messed it up. Sorry. But yeah. Um okay, yeah. Just remember, purines have 2 rings, purines have 1 ring, and then memorize the 5 different nitrogenous bases. Um, yeah. Okay. Also, in this structure of DNA, okay, like basically the bond between the pentose sugar and the phosphate group is called a phosphodiester bond, and that bond is formed by I forgot what it's formed by. It's probably dehydrogenation synthesis. Yeah, it's formed by dehydrogenation synthesis. Okay, that phosphodiester bond, and so that's what bonds this stuff. Okay, and then yeah. Okay, wait. Yeah. Okay, and um and so when you have a DNA, when you're looking at DNA, know that DNA has double helixes, while RNA has only one helix. I don't have like an image pulled up, but basically this is DNA. You have like 2 adjacent strands facing each other. When you have RNA, it's only one strand, and there's nothing facing each other. So basically, with DNA, the 2 strands that face each other are actually opposites of each other. Well, they're anti-parallel. So in DNA, you have one helix. There's the 5 end, the 5 apostrophe end, and you have the 3 apostrophe end. So the thing you always read from 5 end to 3 end. However, one side of the alpha helix is going to be from 5 to 3. The other one's going to go from 3 at the top and 5 is the bottom. So one side you read from top to bottom, other side you read from bottom to top, because you always read from 5 to 3. But one side is going to be flipped around, and so they're anti-parallel. They run in opposite directions. That's something to really keep track of. So when someone shows you the genetic code, it's gonna be in the form of like AGGT88 or something like that, like AGCT whatever. Okay, it's gonna be like that. Okay, and it's gonna always start from the 5 end to the 3 end. However, they're gonna show both strands next to each other, but remember that one strand is always gonna be reverse of each other. Like the complementary strand's always gonna be, you're always gonna read back, like it's gonna be backwards, because it reads from top to bottom actually. So yeah, just remember that. It's pretty hard conceptually to get. I don't know. Just keep that in mind. It shows it here. Like it goes from 5 to 3, 5 to 3. So yeah, yeah. Okay, so this is gene expression. Okay. Oh wait, I do have an image. Okay, yeah. Wait, this is what I was talking about. Okay, yeah. So, with DNA, you have adenine, cytosine, guanine, and thymine. Adenine goes with thymine, cytosine goes with guanine. With RNA, you have uracil, cytosine, adenine, and guanine. Uracil goes with adenine, cytosine goes with guanine. And then with DNA, you have 2 strands, and then with RNA, you have 1 strand. DNA is a double helix, or RNA is a single helix. So yeah. And then the anti-parallel, yeah. Okay, basically. So, what happens in gene expression, okay? So there's some extra information here. All you need to know is that, okay, you have DNA, okay? RNA, in the process of transcription, RNA reads the information of DNA and copies it onto itself, into RNA. So that RNA now contains all the information it needs from the DNA. That was caused by transcription, okay? And then, so RNA is transferred out of the cell, out of the nucleus by mRNA. mRNA is messenger RNA. That's what transfers information. It's a messenger, so it's Hermes. It moves information, across the cell. And so that RNA then, in the process of translation, turns that information it copied into amino acids. It does it by having 3 codons. So a codon is basically just like it's a three-letter code that's unique. Like each amino acid has its own 3-letter code, and that codon is correlative to the genes, like these nitrogenous bases. And then RNA already has the information, so the codon just reads it, and then it's like, oh, this 3-letter code matches with the codon for, I don't know, alanine. So it pulls up an alanine and then it puts it down, and then yeah. So translation is that process. That's basically the very basics of gene expression, where it turns DNA into proteins. It goes, later on we're going to go into really specifics. I think it's the next unit. Next unit is about cells. So yeah. But that's all you know for cells. Oh no, nucleic acids for now. Yeah. Okay. Last concept. I don't know. It's kind of like Campbell's thing where like it just talks about like the future and stuff like that. But basically, humans very much recently in the past few decades, we've been working on we've been working on genomics, which is basically when we look at the genome, and we've also been working on proteomics, which is when we look at proteins. So yeah, the Human Genome Process, the Human Genome Project is basically where we mapped out the entire human genome, and we figured out the entire human genome. We mapped it all out, and then that was really helpful. It helped us do a lot of stuff with like diseases, stuff like that. And then for proteomics, we recently just invented like machine learning models that know proteins, stuff like that. Okay, and then lastly, there's also bioinformatics. So bioinformatics is the use of computer software and other computational tools to handle large data sets, typically with like proteomics and genomics. And lastly, okay, DNA and proteins are kind of not proof of evolution. You can never prove evolution, but they're basically allow us to trace back on evolution. And you can see how all animals, or no, not all animals, all organisms, all life has DNA, and all life has proteins, and it's just like, wow, we all have a common ancestor or something. Yeah. But yeah, that is everything. That is all. That was one big lesson. But we finished the whole unit. We finished the whole lesson. I'm very tired and thirsty. But yeah, remember all that. Have fun studying. Okay, bye.